Cofactors. The discovery of transcription factors and their cofactors are important for transcription regulation. These transcription factors and their cofactors bind the DNA segments to activate or repress the gene expression nearby. Cofactors are often required to transmit regulatory signals between gene-specific activators and the general transcription machinery. There are three types of general cofactors such as 1. TBP, that is Tata binding proteins associated factors, 2. mediator complexes, and 3. upstream stimulatory activity derived positive cofactors such as PC1, PC2, PC3 and PC4, and negative cofactor 1 which is known as NC1 normally function independently or in combination to fine tune the promoter activity in a gene specific or cell type specific manner. Cofactors are required for the transmission of regulatory signals between the gene specific activators and general transcription machinery. Tata binding protein TBP. The TBP is a general transcription factor that binds specifically to a DNA sequence called the Tata box. This DNA sequence is found about 30 base pair upstream of the transcription start site in some eukaryotic gene promoters. It is estimated that only 10 to 20% of human promoters have Tata boxes. Tata binding proteins associated factors is a part of TF2D multimeric protein complex that in turn makes up part of the RNA polymerase II pre-initiation complex. The TBP is an unusual protein which contains long string of glutamines in the N terminus of the protein, which binds the minor groove using a beta sheet. It also modulates the DNA binding activity of the C terminus, and modulation of DNA binding affects the rate of transcription complex formation and transcription initiation. The first Tata binding protein associated factors, that is TAF, was discovered in Drosophila melanogaster and humans. These proteins were identified as biochemically stable compounds and distinguished by a number which indicates their predicted or apparent molecular weight. TAF proteins adopt a histone like fold. TBP is probably not the only protein involved in positioning RNA polymerase II, but it is involved in double strand separation of DNA known as DNA melting by bending the DNA at 80 degree. The AT rich sequence to which it binds facilitates easy melting. Transcription coactivator mediator complexes. The transcription coactivator mediator complex of the transcription mediated by RNA polymerase II bridges transcriptional activators, coactivators, general transcription factors and RNA polymerase II to repress or enhance the formation of the transcription pre-initiation complex. It also participates in the formation of several functional complexes such as the activator recruited cofactor, cluster recruited to nuclear receptors and stimulator of RNA binding MET cofactor complexes that particularly works for activation of transcription by hepatocyte nuclear factor 4, another SRB MET cofactor complex, NAT complex that repress the activation of transcription by RNA polymerase II, cofactor required for specificity protein 1 transcriptional activation that is required together with TAF2s to activate the transcription factor specificity protein 1 and positive cofactor complex 2. Mediator complex subunit 1 is a component of mediator that associates with the nuclear hormone receptors. With the mediator kinase molecule, mediator forms the thyroid hormone receptor associated protein complex that repress transcription. With the subunit MED26, the mediator complex known as positive cofactor 2 activates transcription. Upstream stimulatory activity derived positive and negative cofactors. The upstream stimulatory factors are homo or heterodimers that pertain to the basic helix loop helix leucine zipper class of transcription factors to bind the E box element. Positive cofactors 1 corresponds to PARP1, that is poly ADP ribose polymerase 1. Positive cofactors 2 with MED26 mediator, positive cofactor 3 to DNA topoisomerase 1. The negative cofactor 1 NC1 corresponds to HMGB1, that is high mobility group box 1 protein. In addition, other cofactors such as TAF1, BTAF1 and negative cofactor 2 can also modulate TBP or TF2D binding to the core promoter.